So, good morning, everyone. So, it's a real pleasure to welcome you to this first session of the second half of the conference. On this educational session of effects of structural variants on the 3D genome, we have an exciting duo of speakers. I am Nicolas Chatron from the Lyon University Hospital, and I'm chairing this session with...
Hi, I'm Sid Banka from University of Manchester, and it's a great pleasure to invite our first speaker to the podium, who's Dr. Malte Spielmann. He's Director and Chair of Human Genetics at the University of Lübeck and Research Group Leader at the Max Planck Institute for Molecular Genetics in Berlin. Over to you, Malte.
Thank you so much for coming out so early. It's really an honor to be here and to talk to you about structural variants in the 3D genome. And I want to share with you some educational stuff today, and then also show a few unpublished things, but let's start right from the beginning. So I'm, like many of you, a clinical geneticist, and I'm interested in solving my patients. And I'm very interested in rare disease, and these are some of my patients, and I know some of you in the room probably can look at the tip of the nose of one of those patients and know exactly where the mutation is. I was never really that good, and during my education as a clinical geneticist, I always dreamed of a technology that could do the trick for me. And then, roughly 10 years ago, this paper came out. And by some people here in the room, it's still a legendary paper by my dear friend Christian Gillissen. In 2014, they did whole genome sequencing and could solve roughly 60% of all patients with severe intellectual disability. And when I saw this, I thought, that's my dream. Let's do that. That's awesome. It took us 10 years to get even started, and I moved to Schleswig-Holstein, which is the most northern state in Germany. And there's two cities, Kiel and Lübeck, and we were very proud to, 10 years after Nijmegen, to start genome sequencing in the clinic. And I want to share with you just 2 slides on how we do this and then how we fail in solving all our patients. And so I think in general, whole genome sequencing, and I'm talking about short reads here, could really do the entire trick. You get your sequence, obviously, but you can also reconstruct the karyotype if you're good. You get the deletions, duplications, and in some cases, even the repeats. So you should be able to solve all our patients. And when we started doing this in Schleswig-Holstein, we were able to solve roughly 42%. So I was very proud and walked around and said, like, well, we are a cool institute, we do whole genome sequencing. But then when I talk to parents, then they say, so what's wrong with you? Like, you sequence the entire genome, but you only solve 40%? So then I felt very insecure, and I thought maybe we're doing something wrong. So I looked into the literature and came across the 100,000 genomes pilot paper, and they were only able to solve 35%. So I thought, at least we're in good company down there. And so the question here is, and that's what I want to talk about today, what are we missing? Like, if we are looking at maybe we're looking at the genome the wrong way. So, and since we're talking about structural variants and 3D genome architecture, obviously I'm going to move that direction, but let's start with a very simple slide that is very old, but still makes a lot of sense to me. So how do we actually currently look at our diagnostic patients? So if you have a patient that has intellectual disability and you do some kind of test, you find a deletion, this one here, and then you obviously look at the genes inside. If this gene here, for example, causes intellectual disability, case solved. Very easy. But what happens more frequently than I like is that you find a variant and none of the genes make any sense. Or worse, now with whole genome sequencing, you find variants that are so small that there's no gene inside at all. And then what are you going to tell the parents? Your patient has a deletion of nothing, right? That's not going to do the trick. So I was wondering what else could there be, and this slide already imposes that there's not just genes, but also something in between. So, and that's why I always show this slide, and when I do this, I still think it's a scandal, right? You know this, but our genome consists only of roughly 2% genes, so protein coding genes, and 98% junk DNA, so non-coding sequence. And I don't personally like the term junk DNA, but prefer the term gene deserts because it kind of implies that you can still discover something there, like Indiana Jones. But I can tell you, you can also get lost in the desert. But what's the big problem with the non-coding genome? That we don't understand the code. We simply know very, very little. But let me talk you through how little we know and how we can use this to diagnose our patients. So in each of your cell, you have roughly 2 meters of DNA, and they are tightly packed into the nucleus. And if you look at it, then it's not completely random like spaghetti, but there's actually the chromosomes have certain territories. And if you change them, this causes trouble. And then if we zoom in even further, we see that the chromosomes actually, or the DNA actually forms loops between non-coding elements and genes. But because this is so complicated, I like to make it much more simple and show it to you in another way. So I told you now that we have genes, and there are very few of them, and then we have non-coding elements like this, and we don't understand the sequence, but if we take a sequence like this and make a transgenic mouse with a blue staining on it, then we can see this sequence here seems to be an enhancer, like a switch, that drives expression in the limb. And then we have other enhancers, like this guy, that drive expression in the brain. And in order to keep them separate, there's something called boundary elements. So how does this enhancer now drive gene expression? And this is done by DNA looping. And this happens in 3D. So basically the DNA with the enhancer bound by some transcription factors finds its target gene. And this is tissue specific. So in the eye, you get a contact and expression, but in the brain, it might be different. So the cool thing is you can actually measure this. This is not just an idea. And how we do this is by chromosome conformation capture. And there's different flavors of it, but just stay with me. So I told you here, you have this looping. So what you can do then is to fix the cells and then digest off all the rest of the genome, and you have your enhancer stuck to your target gene. And this is where we start here in the high C protocol, or 4C. Basically, you have them stuck together, so you have your favorite gene and the maybe unknown enhancer sequence. You digest off the rest, and then you make a circle, and then you can make 2 different flavors of this. One is called 4C, that you see down here, and there you have your favorite gene as a viewpoint, this guy, for example, and then you see those tissue-specific interactions. So this might be an enhancer, and this might be an enhancer. But since we live in a high-throughput world, we do this at higher throughput, and this is called high C. It's developed over 15 years ago. And we see here that you see chromatin interaction in these regulatory hubs. And in order to explain this a little better, what does it mean? So here you have one gene expressed in the neural tube, and that has several enhancers, and they keep looping towards that gene, and that's why we see this heat map. And then you have a neighboring gene expressed in the embryonic limb that has its own enhancer. And how does this looping happen? And this is a bit complex, but actually quite dynamic. So basically you have the cohesin ring, this is a protein, and that kind of sucks the DNA through with its enhancers. And then here you have this loop where there's interaction between the enhancer and the target gene. And then it keeps looping towards these CTCF sites. And once it reaches it, this pops off and then goes back into the same loop. And that's why you see this is a constant hit and run process. It's very dynamic. But when I first saw this in 2012 at the Royal Society, I thought, what does it have to do with my patients? And I look like some of you out there. But then I thought, what about this guy? What about this boundary element? So how does this enhancer know that it's only supposed to drive that gene and not the neighboring one? And this is due to these boundary elements. And I wasn't just thinking about any patient, but a long time ago, now 10 years, I had seen these patients in Berlin, and they have a brachydactyly, they have short fingers. And they had deletions of a gene called FRNA4, and we had actually made a knockout mouse. It took a long time. The mouse was completely fine, right? It was great for the mouse, not so great for us. But then with the knowledge now of the topologically associating domains, this is what they're called, then we thought maybe this deletion, it actually also removes the boundary element here. So maybe this could have something to do with it and help our patients. So we came up with the following hypothesis. So in the wild type, you have one TAD here with its own enhancers and a neighboring TAD here. And now if you delete the boundary, what would happen is that you get a process that we call enhancer adoption, or nowadays we call it enhancer hijacking. So this enhancer here, which usually drives gene A, is now able to drive the neighboring gene, and this is then expressed where it should be, but also in a region where it should not be, in the limbs. And this was a great hypothesis, but no one wanted to believe us. So we, long time ago, made some knockout experiments using CRISPR-Cas where you can make deletions, inversions, and duplications. And this is what it looks like. So I told you, in the wild type, you have a gene over here that is not expressed in the limb. The gene was called PAX3, and only a little bit here in the muscle. And if this is true, you get a beautiful hand in the human and in the mouse. And then if you delete it, the boundary, suddenly this enhancer hijacks the neighboring gene, and indeed you see here that there's massive misexpression causing exactly this phenotype. And this was the first time that in congenital disease, these TAD boundary deletions could be shown to cause disease. So we were very proud, and over the years published lots of other stories on this. For example, you can also have a translocation or an inversion, and the same thing happens. So if you take this enhancer and invert it over here, then you also get trouble, because now this enhancer will hijack that gene, and this one that gene. At the same time, its original enhancer is lost. So you get a complex phenotype of enhancer hijacking and loss of function. And you can even do this for duplications. We, at the time, called it neotad formation, once again based on very rare cases. So this is, if you have a duplication here, you think, well, there's a higher dose of genes, but also you change the 3D genome architecture of the locus. And how do we change it? You basically, if you duplicate this in tandem, you have suddenly a new TAD where this fragment, which is supposed to drive the blue gene, suddenly is in a compartment with the other one, and you get enhancer hijacking again. So we showed this once again on skeletal stuff, and after a few years, we actually got invited to write on big journals, and after first people laughed at us, I was very proud. And this is a father moment, so I took this magazine home and showed it to my son. And then he said, "Oh, Dad, finally I understand what you're working on. You work on pyramids." And when I told him that I'm just a geneticist, so he completely lost interest again. But I think what's the bigger picture here is that this mutational mechanism is now widely being recognized in cancer. And for example, our colleagues at the German Cancer Center published at the same time that enhancer hijacking is actually a quite frequent mutational mechanism in medulloblastoma, and also for leukemia and other cancers this has been shown. And quite easily this is understood because cancers just mutate like hell, and we're going to hear some cool stories about that in the next talk. So I could stop here, but this is basically just a review of what has been done in the field, and now I want to show you some newer stuff and come back to more specific problems of genome sequencing. So I told you about deletions, if you take those boundaries away. I told you about inversions and duplications. Now, one thing that is cool now with genome sequencing, in particular with long reads, you can also now map insertion breakpoints. And this can come in handy and has something also to do with 3D genome architecture. And I want to share one story that's also published, but very dear to me, because this is a great collaboration with my good friend Elisa Giorgio from Italy. And she, a few years ago, when I was at the SPC and the ESHG, said, well, we have this case that is a very tragic one. And you see here, this is a baby girl. She was born, and at the age of 10 months, the parents noticed that her arms are very stiff and her entire body. And when they did a CT scan, they could show that her muscles turned into bone. A very similar phenotype to ossificans progressiva, but more severe. So at the age of 5, her entire body was basically a skeleton. And then she tragically died, and they couldn't find out what was going on. They had done exome, they had done arrays, so we at the time did something very cool, genome sequencing, and were able to identify reads that mapped from chromosome 2 to chromosome X, smelling a little bit like an insertion or translocation. And then Elisa, because she is a great geneticist, said, well, we could have done that much cheaper using cytogenetics. And indeed, when they had found that duplication earlier in the array, but they didn't know that it was translocated into the X chromosome. And if you pay attention down here, then you see this is chromosome 2, and then here there's the insertion on the X. So how would that change 3D genome architecture? And for that, we got some cells from the little girl and performed Hi-C. And this is a Hi-C map from fibroblasts. It's not as beautiful, but you can see here that the gene called ARGAP36 has its own TAD. And maybe you focus on the cartoon here because it's easier to understand. And the insertion breakpoint is exactly in this TAD. So when we did it from the patient, you could see that there's some kind of gap here. And that's why when we reconstructed the genome, this is what it looks like. So this was the wild type, and here you have the insertion. And then when you focus on the sides, thinking about position effects or 3D genome architecture, then you can appreciate that there were several new contacts formed between the ARGAP36 gene and enhancers located actually in this inserted fragment. And normally they drive a gene located on the chromosome 2, but now they can hijack ARGAP36. And because there's another gene on the other side, we performed RNA sequencing on these patient fibroblasts to see what would happen. And we saw that this gene here was not changed, but that the ARGAP36 gene, this one here, was massively upregulated and smelling a little bit like enhancer hijacking again, so position effects. And I don't have time to show you the entire paper, but what we did then, we could not make a mouse model because this fragment was several hundred kb, but we showed in vitro that the overexpression of ARGAP36 can actually turn muscle into chondrocyte-like structures in a dish, indicating that this is indeed the mutational mechanism. For me, this was, first of all, a very tragic case, but at the same time also this is not my work, is quite interesting how you can turn one tissue into another, and this is exactly how we think of misexpression and position effects. Good. So I talked to you about bones, just because I like bones, and I had to realize the hard way that not everyone in the clinical genetics community cares so much about fingers and bones like I do, but most people work on intellectual disability. So people always told me after my talks, it's nice with your fingers and stuff, but can you talk about some real interesting disease? So I want to share with you some unpublished work about neurological disorders, also caused by structural changes in the 3D genome. And this is once again a collaborative story with Elisa Giorgio, and she, many years ago, identified, or this has been known, that duplications of the gene called LMNB1 cause a disease called adenoleukodystrophy. And this is well known and has been extensively published, that people think of this as not of position effects, but clearly as gene dosage. If you have a duplication of LMNB1, you get this generalized demyelinization of the brain, causing adult onset leukodystrophy, a terrible disease. But what Elisa and I discovered a few years back is that there were deletions right next to LMNB1. So not touching the gene body, but deleting a region that was upstream of it. And if we look at the phenotype, also they have leukodystrophy, but the cerebellum was spared, or at least comes later in development. So we thought that smells a little bit like position effects, but how can we study it? And I always backed away a little bit from neurological disorders because you can't model it very well in the mouse. Because some people tell me they can see if the mouse is autistic and is very sad or dumb, but in a mouse I'm not so sure. So I wanted to test this in a better way. And we use single-cell RNA sequencing. So I told you that here the cerebellum is spared, but now Varun, a very clever postdoc, and Jana, a student in my lab, made mouse models, and the hypothesis was, once again, enhancer hijacking. So in the wild type, LMNB1, or in the classical case, it's duplicated, and you get gene dosage effects. But if you delete the upstream fragment, you get classical enhancer hijacking. So the enhancers of the gene over here can now hijack LMNB1 and drive this tissue-specific overexpression. And we made several mouse models for the classical dupe and for the newly discovered deletion and the wild type. And because I told you it's difficult to look into a mouse brain, we performed, first of all, high-C experiments to see if there's really enhancer hijacking going on, and extensive single-cell analysis, and also some MRIs, which I will not share today. And this is the locus and this is what it really looks like. So bear with me here. So you have LMNB1 sitting in its own TAD, and then you have a neighboring large TAD, which has a bit of a subTAD here, where there's lots of enhancers that normally drive that gene. So what you can do from high-C data is to reconstruct it using virtual 4C. And what it basically does is that it shows you, here you have our favorite gene, and these are all the genomic interactions. So that basically respects these boundaries and doesn't go into this TAD over here. What happens now in our patients if you have a deletion? Then this boundary is gone, this entire fragment, and you get massive interactions with these enhancers here. So that smells already like enhancer hijacking, right? There's ectopic interactions. And what does it now mean to our patients? Here we did a rescue. So basically we wanted to see, is this specific and could this rescue our phenotype? So we took a boundary from somewhere else and knocked it back into the mouse. And then you see this contact is completely abolished, right? And the phenotype is actually rescued. But more about that later. Good, so on the chromatin level, enhancer hijacking is very clear here. And now to the brain. So we took the entire brains of those mice and sequenced them, and then I guess many of you are now familiar with single-cell RNA sequencing. Then you get basically these clusters that represent the main cell types, and each dot represents the transcriptome of one cell. Then you can give them nice names, but the first thing that we wanted to see, if indeed there's enhancer hijacking going on, what does it mean for transcription? Is LMNB1 now hijacked? And that's why we focus here on LMNB1 and the wild type. So this is the chromatin context, and you see in the wild type, this is what it looks like. It respects the boundaries. And if we look at expression of LMNB1, it's mainly in these cell types down here. And these are early neuron precursor cells. And then we look at the neighboring gene, GRAMD3, and this shows a very distinct expression pattern up here in the glial cells, so very different. So now what happens if you remove this boundary and also the gene? Then we thought these enhancers that normally drive GRAMD3 should hijack LMNB1. And we can see this exactly on the RNA level. So first of all, in the deletion, you see the spread of contact. So there's interaction. And at the transcriptional level, you see that suddenly LMNB1, which should look like this, is massively overexpressed in these glial cells where it shouldn't be. So this, once again, is a clear indication of enhancer hijacking. But what does it do to the cells of the brain? And that's the cool thing about RNA sequencing. You can understand now, almost like a microscope, how the cells behave, and are they in good shape? Are they changing their identity? So I will not go too much into detail, but 2 years ago we published an algorithm called Loch Ness that gives you the ability to understand RNA sequencing data a little bit better, in particular in mutants. And we call it, it's kind of a mutant score, and normally you have wild type cells, because we always have a wild type also sequenced. And if there's no phenotype, then you see that there's always a wild type cell next to a mutant cell. This would be a mutant score 0. But then there's region where there's more mutant cells. This would be called mutant score high. Or there's a region where there's lower, there's an absent cells in the mutation. And because I think this is difficult to understand, I told at the time Fanny, the computational scientist, just make it understandable for me as a doctor. Color it in blue and red, then I see where I have to look. And so this is what I will show you now. So now we are looking at the mouse brains, and this is the classical duplication, and then we see red, there's some losses, and blue, there's some gains. But what does it mean compared to the the deletion? And we see that it looks actually very similar. For example, if we look at these guys here, these are indeed the cortex neurons, and they seem to be gained in both, like an overexpression, even though, and this causes exactly the phenotype that we see in the patient, in the cortex, this generalized leukodystrophy. So this is nice, but are there also differences? And you can appreciate that, for example, here it's blue in the dupe, and in the deletion it's red, so here's a loss. And this is exactly where we see the difference. These are the neurons of the cerebellum, and you can appreciate that there's a difference, just like in our patient, where in the dupe it's, there's overexpression, and in the del there's not much happening. So we are still putting the pieces together, but I just wanted to give you this as a slight outlook on what else we will do in the future. I think it's pretty exciting times. Good. Now, a short break. I told you now about the TADs, and that if you remove a boundary, you get trouble because you get enhancer hijacking, and told you different flavors if you have a DUP, a DEL, inversion, and you can even see that in neurons. But is it really just all TADs? And obviously the answer is no. And I want to now tell you a story about how also coding mutations can change the way how the chromatin is folded and cause trouble genome-wide. And this is once again a very clinical story. And a few years ago, actually a long time ago, Martin Menzer and I, and when I was still in Berlin, got a sample from Hong Kong of this child, and it is a very syndromic fetus that has skeletal abnormalities of the lower limbs, of the hands, and we performed genome sequencing, short read based, and found a de novo mutation in a gene called HMGB1. It was a frame shift, so I thought it's disease causing, but the problem was the gene is already taken. Mutations in this gene cause mild intellectual disability. And this is not the problem of the child, so we didn't know what to do. And then I think sometimes this happens, we just didn't do anything. This was in 2012, we just let it sit in our drawer. And then many years later, simply because we didn't understand it, but many years later, I moved to Kiel and Lübeck, and one of the first patients that we saw there was this one, and Almut Kalibe, the clinical geneticist working with me, identified this patient also prenatally, and it had the exact same features, so absent tibia and polydactyly, and also some facial feature. They actually also have an intersex phenotype. And then together with colleagues from Berlin, they realized very quickly that this is actually a recognizable syndrome called brachyphalange polydactyly tibial aplasia syndrome. Long word, sorry. We say BBTAS. And so we collected patients from all over the world, and it's rare, so we found 7. Right? And then we did whole genome sequencing, and to my surprise, or all our surprise, we found the identical de novo mutations in the last exon of HMGB1. But I told you, the gene doesn't make any sense because it causes mild intellectual disability. So then we were sitting together, and I got back in my molecular genetics knowledge and thought, wait a minute, what happens if you have frame shifts in the last exon? It doesn't always cause nonsense-mediated decay, but sometimes you actually have a stable protein. And so we looked at it more carefully, and this is actually an intrinsically disorganized region. So for us, the worst, because we don't know what they mean. But indeed, normally in all species, the gene stops here, but here we had an acidic tail that now created a new gene. And what does HMGB1 do? It's a transcription factor. So now I thought, wow, we have kind of a gain-of-function variant now that can go wild in the genome. And now coming back to 3D genome architecture, what does HMGB1 do? And HMGB1 is indeed one of those transcription factors that mediates enhancer gene contact. And I told you it looks like this, but I was lying. I thought it looked like this, but I talked to my friend Dennis Hinz at the Max Planck in Berlin, and he said, Malte, you're very naive, it doesn't look like this, it looks closer to this, right? It's an entire hub. It's hundreds of transcription factors coming together. And the cool thing is you can even see this under the microscope, and this is called phase separation. And it's once again works in a hit-and-run fashion, but these condensates form in the nucleus and then drive gene expression. If something goes wrong, you get in trouble. So coming back to our patient, what would that mean if we now have a mutation in HMGB1? So first of all, we wanted to know what does it do? And HMGB1 is, we put a GFP on it. This is work by a PhD student of Danish. And you see it goes into the nucleus, but you don't really see anything. Normally, it's just there. So but if you have a mutation, it would get more sticky, we thought, and then maybe we would see those condensates. And indeed, this is exactly what happens. In the mutants, you get this internuclear condensates, and transcriptionally, the cells go wild. This basically means you have an enhancer or many enhancers stuck to their target genes, and they keep firing, right? So we thought this was super cool, and sent it to a big journal, and they said, this is so lame, like this is super rare, you've just solved 8 patients, we don't care. So we were very sad and thought, how can we maybe make this a bit sexier or make it more relevant? So we did a trick. And we just went online and looked, maybe this is a more common mutational mechanism, not just my disease, but maybe yours. And we just downloaded over 5,000 genes that have this disordered tail, and then filtered for frameshift mutations in the very last exon. And we found over 600 different genes where this is the case. And then obviously reviewers don't like statistics, they want you to prove it. So we then tested some of them. And here I show you just a few examples. So, for example, this is the closely related HMGB3, so the brother. And once again, in the wild type, you see a bit of green, but in the mutant, you see those condensates, and they go crazy. And I will go not through all of them, but let's just focus, for example, if people interested in cancer. RUNX1, actually, you have a bit of phase separation already in the wild type, but in the mutant, it gets much more severe. So it's basically a gain of this contact. And I thought this was cool, and when I presented this to a clinical crowd at a meeting in Bertinoro, Han Brunner came to me, and actually Alex Heuschen, and said, I know another syndrome that's exactly like that. And I will, because I like bones, I also thought it was interesting. Robinot syndrome was published many years ago as causative in mutations at DVL1. And interestingly, only mutations in the last exon, frameshift, cause this phenotype. And the rest of the gene is highly polymorphic. So in GNOMED, you find lots of variants. So we thought maybe this is the same problem. And indeed, when we took DVL1 in the wild type, it doesn't even go into the nucleus. But when we took the mutant version of it, this frameshift with the acidic tail on it, you see these 2 condensates, and again, transcription goes wild. So we indeed believe that this is a much more common mutational mechanism than previously thought. We also found this now in differences of sex development, but in the interest of time, I will skip this guy and come to the last part. So what else are we missing? I told you now about TADs and disease. I told you that even coding mutations can change the 3D genome architecture and then in turn cause transcriptional problems. So what else is there? And I think here I'm trying to set the stage also for the next talk. I think at this meeting it's now clear that we're currently only sequencing, when I talk about whole genome sequencing, I'm lying, because we're just sequencing a little bit the genome. And these gaps here have been filled by an international team of the Telomere to Telomere Consortium and Evan Eichler and his gang. And now many people, we saw this in the What's New session, also use it clinically. And how does it work? I will just be brief here. We in the lab use hi-fi PacBio sequencing, but there's different flavors of it. The bottom line is that you get much longer reads and then can build a much higher and better reference or patient genome than previously thought. And the idea here is that this could help us understand mutations, in particular in the non-coding region, which seems to be much more tricky to capture, better and faster. So when I first learned about PacBio sequencing, I thought we need to find a showcase. And I had moved to Lübeck in 2020 and got to have the privilege to work with a great attending, a geneticist interested in neurological disease. And he had a family, which for German standard is very large, affected by what we call spinocerebellar ataxia type 4, SCA4. And this had been mapped to chromosome 16 22 years ago, so ages ago. But we just couldn't solve it. And we tried everything. We tried, at the time they tried positional cloning, they tried exome sequencing, short-read genome sequencing. We just couldn't solve it. So I thought that's the perfect example where we have a locus, we don't get disturbed by all those variants. We just look at what's inside. And we did 2 PacBio genomes of these 2 guys here, and immediately found a coding repeat in a gene called ZFHX3. And people didn't believe us because they thought, well, we've sequenced this gene, and this repeat is a GGC repeat. It's so difficult to detect using any other technology that we had to confirm it using nanopore sequencing, because the short read, any technology just wouldn't work. We couldn't even get a PCR up and running. So I thought that was pretty cool. And at the time, I invited Tobias Haag, the head of molecular genetics in Tübingen, which is the largest institute in Germany, to our institute in Lübeck. And I told him, we are this cool place, we do long reads, and we just solved the case. And then he said, but it wasn't SCAR4, was it? And I'm like, and they found the exact same coding repeat 1 week earlier than us. And so then we sat in shock for a minute and just decided, okay, let's join forces. And we, in the end, published a beautiful paper last year. But at the same time, 3 preprints came out of other people in the world also finding that same repeat. And why am I showing you this? Because I think this just nicely shows that long reads can actually show us things that we haven't seen before. And I'm not sure, we can discuss it maybe also with Tobias in a bit, what is it really the tip of the iceberg? So if we now do long reads, will we just solve all our cases? I'm not sure. And we, once again, in the highlights session yesterday, or the day before yesterday, we saw that maybe we will not solve all our cases, but every one of you probably has one of those families where long reads might actually make a lot of sense. Good. Now I come to my conclusion. I hope I could convince you today that the non-coding genome is really the next frontier. And I always show this slide, which I actually stole from a colleague of mine. But if you were an alien coming to Earth, I would highly recommend you to go to one of those places where there's lots of light. You're going to meet people, right, here in Italy or in Germany or in the US. But that's not all, right? And this is how we've done diagnostics so far. We just focused on the genes, and that makes perfect sense. Everyone has been extremely successful. But now, more recently, people got interested in crazy places, right? And this is those gene deserts that I talked about. And let's focus on Greenland for a little bit, just because everyone is now interested in Greenland. And I thought there was nothing there. But my dear friend, Nikki Whiffen, and you all know this now, just identified non-coding mutations of this RNA causing the Renew syndrome. This was only published last year, and no one saw it coming. So it was really in the middle of nowhere. And just through the power of whole genome sequencing at large scale, they were able to identify this. So I think it's worth going to those deserts, finding new stuff, because it will actually help our patients. And also on a different note, also therapeutically, I think we have to move to the non-coding part, also in cancer. Because, for example, the only approved gene therapy using CRISPR for sickle cell anemia and beta thalassemia doesn't actually target a gene, but it targets a non-coding element, one of those enhancers I told you about. So I think if we go that way, and there's now national initiatives going on, obviously the 100,000 Genomes Project, but even we in Slow Germany are going there, there's going to be much more. And it's going to be exciting, and it needs to be done together, right? No one can do it alone. With this, I summarize. I hope I could convince you or talk to you that structural variants really need to be studied in the 3D genome, because they change how the architecture works and can rewire enhancer promoter contacts. Then I would strongly urge you that you need to think beyond the exome, and there's an entire session on the Renew syndrome, so I think everyone agrees now. And I hope that you also agree that the non-coding genome is indeed the next frontier of human genetics. And I've been walking around for 10 years saying that. I'm just happy that finally someone found those non-coding mutations. Good. Now, this is an educational session. I wanted to highlight, I showed you some stuff and we did lots of experiments, but what does it mean for you? Can you do that with your patient? And the answer is yes. because it's all publicly available. You don't have to make those mice. You don't have to do Hi-C. If you simply go to the UCSC browser, you find the TADs, you find in the Vista browser, all the enhancer data are there from many different sources. You can go to the Hi-C browser. It will show you your favorite gene and the locus. And there's even more sophisticated algorithms, if you know a little bit of bioinformatics or you ask your friend, that can now predict enhancer promoter intacts in pathogenic situations for you. For example, I like an algorithm called Postre, but also DB Regulome. And if you're interested, we just in the process of publishing a new review where we also included an algorithm, basically how if you have a structural variant, a deletion or duplication, how you can go through that algorithm and then solve it and see if it's indeed changing 3D genome architecture. With this, I would like to thank my team because I'm literally just talking about this stuff. I don't do any work, unfortunately, in the lab anymore, but they are doing it, and I want to thank really everyone, but particularly Varun and Jana, also Martin, but also Christian. I also showed some work for Moira, and obviously the funding sources. And I also want to thank our clinical team. They do great work. And last but not least, I want to show you this. This is our research building in Lübeck, and we're very proud of it. This is where I work and my team. We sit on this floor, so it looks very fancy. But if you take a very careful look, then you can see that there's an empty computer or an empty bench where you could work. So if you like what you saw today or you want to collaborate, then please send me an email. We are recruiting. I would be happy to talk to you. I hope there's some questions. Thank you, guys.
That's absolutely fabulous, Malte. Real tour across the developments in the field and also looking into the future. Are there any questions? Please come to the microphone, introduce yourself, and ask your question.
Hello, this is Pino D'Ambrosio from Rome. I think it's very clear that genome sequencing, Hi-C, and long-range sequencing can help patients directly. But we're having a hard time finding fundings for that. So I'd like to ask you the German experience about who pays for these exams when they directly help patients.
Yeah, thank you so much. So in Germany, we are in the lucky situation that our health minister a few years back decided that he wants a genome sequencing project. I think this was more like a prestige thing. But we, as an entire community of rare disease and also cancer geneticists, convinced the government that this should be available to all patients. And now we started a pilot project for 5 years that every patient in Germany can now get their genome sequencing if they fulfill certain criteria. So in rare disease, they have to be at an academic center, and in cancer, they have to gone through the standard therapy. And if they fail, then they can get the tumor and the genome sequenced. And this pilot project is funded well and will also enable us to do some functional workup. Right? And this will not be super fast, but we hope to show by this and come back to our government after 5 years that indeed, first of all, genome sequencing can help us, and I think there's very good evidence for rare disease. We will find more, but that will also help us to show that in academic centers and through international and national collaborations, we can actually make better diagnoses. And getting the twist to cancer, I think also that if they venture into the non-coding world, that they will find new druggable targets. That's the idea. And I know there's other national initiatives, and I hope that in Italy this will also happen soon.
Hi, Wouter Bossuit from University of Leuven. So I was wondering, the annotation of CNVs in the clinic is still a problem, and boundary elements, as you say, are a potential explanation for some of those. So are there tools available to annotate the CNVs with the knowledge of boundary elements locations present?
Yeah, thank you so much for that question. And indeed, there are tools, and these boundaries or the TADs are no secret. So that's why I said if you just look at the UCSC browser, you will see them. And the fun thing, and this is, I didn't say that, 10 years ago, when the TADs were first identified, the thing that struck most of us the most is that they're actually quite stable between cell types, even between species, between mouse and men. And that means those boundaries also are quite cell type stable. So, and while the interactions within the TADs are very cell type specific. That means if I'm happy to send it to you, but it's also out there. You can, for example, this is what I do. If I look at CNVs, I just have a boundary track always running in the background, but you can also do this bioinformatically. For example, I know that also now some of the commercial tools for genome analysis try to include those boundary elements. And then there's also some more advanced tools, like the one I called out called POSTRE. It's a paper where basically you can upload your CNVs and it will try to predict, do the whole trick, like where are enhancers, where are potential target genes, and where would they come together, and then it will rank them. But also, once again, I'm happy to share any information and work together. And if someone finds one of those boundary deletions and isn't sure whether this is a good one, then I would always recommend use your clinical knowledge, right? If this is a smoking gun, like the case I showed with LMNB1, directly next to the target gene, then that's a good one. If it's in the middle of nowhere, then you have to convince your student to make a knockout mouse. I'm not sure. It might be a suicide mission. But overall, there's tools available. Yes.
Thanks.
Thanks very much, Malte. I think we can give you a huge round of applause.
Thanks so much for it.